A scientist was planning to compare the effectiveness of the antibiotic mixopyronin on two different species of bacteria. Figure 18 shows the equipment the scientists can use: agar plates, two species of bacteria, filter discs of the antibiotic, a spreader, and an incubator. Describe how the scientists could determine the effectiveness of mixopyronin on the two species of bacteria. So this is how do we use this equipment to test the effectiveness. So what we'd want to do first of all is we'd want to infect the agar plate with the bacteria. So we want two different agar plates and two species of bacteria. So we want to use the spreader to spread one species of bacteria on one agar plate and another on a different agar plate. We then put the filter discs in the agar plates, maybe put 4 in each, and we put them in an incubator to keep them at optimum temperature for growth of bacteria. And then we would see which one has killed the most bacteria, because around each disc you should get a zone of clarity, a zone of inhibition, where bacteria have not grown, they've been inhibited from growing. And the one with the biggest zones of inhibition would be the most effective, or would show which bacteria species is most affected by mixopyronin. So what you'd say here is spread the bacteria on different agar plates and add antibiotic discs and incubate plates. Measure and compare the zone of inhibition. So remember, that's an important term. If you've got yourself a agar plate and you've got a disc there, you'd expect to get a zone around it where you get no growth, because that is where the antibiotic has killed the bacteria or stopped it growing, and everywhere else you're going to get bacteria growing. So that's a zone of inhibition. Mixopyronin inhibits bacterial RNA polymerase. Explain why the antibiotic mixopyronin can be used to treat bacterial infection in humans. So RNA polymerase will basically stop RNA being produced. And RNA is important for making proteins. So if the bacteria can't make its own proteins, it stops it growing. So explain why the antibiotic can be used to treat bacterial infections in humans, because it only affects bacterial RNA polymerase, RNA, and not human RNA. So basically, RNA polymerase is an enzyme that makes RNA, which will then go on. The RNA was then used to make proteins. So you'd say antibiotics prevent bacteria reproducing, doesn't affect human cells, that's really important to say. Prevents production of bacterial messenger RNA. If you don't know what that means, you should look at the tutorial on protein synthesis. Preventing transcription, that's the part that produces messenger RNA. So there's no protein synthesis. So we just got the idea that the antibiotic prevents bacteria reproducing because it can't make proteins involved in reproduction or necessary for reproduction. It's specific to bacteria, it doesn't affect humans, and prevents transcription because there is no messenger RNA being produced. If this doesn't make sense, as I said, watch the tutorial on protein synthesis, because that could easily be a challenging 6-marker. Infections can also be caused by viruses. Describe the lytic pathway of a virus. Lytic means to break down, so the pathway in which a virus breaks down a cell, and how this causes the spread of infection through a population. So it's not just about describing the lytic pathway, but also how it's spread through a population. So it's quite a grand question, multi-layered again, as these 6 markers often are. So this is how a virus basically works. It lands on a host cell. It injects its genetic material into the cell. The genetic material uses the cell's own machinery to manufacture the parts of the virus, which it then assembles into new viruses. So the genetic material will replicate. It will manufacture or get the cell to produce parts of the virus. The virus is assembled, and the virus is burst out the cell, and they find other cells to repeat this process. And then obviously these viruses can be transmitted through bodily fluids and so on, which can be passed through a population. So how do we write this? Viruses cannot replicate outside a host. So the idea is that's why they're not alive. We don't class them as a living thing. They require a host cell for replication or reproduction. They bind to a host cell, as I've shown you here. They insert their genetic material. They use cell's machinery to produce viral proteins and nucleic acids. So that's the posh way of saying parts of the virus. So viral proteins and nucleic acids, in other words, genetic material, nucleic acids like DNA and RNA. The components assemble into new virus particles, which cause cell lysis. That means the cell breaks down. That's what lysis is. Spread. So that deals with the first part, describe the lytic pathway. Virus particles leave host through body fluids, spread through airborne droplets or direct contact, allowing spread to another host.